Salmonella. Salmonella are gram-negative, flagellated, facultatively anaerobic bacilli possessing three major antigens, H or flagellar antigen, O or somatic antigen, and V antigen. O antigens occur on the surface of the outer membrane and are determined by specific sugar sequences on the cell surface. V antigen is a polysaccharide capsule that surrounds the O antigen, thus protecting the bacteria from antibody attack on the O antigen. The Salmonella infection cycle starts after the ingestion of microbes. Through the stomach, the bacteria reach the small intestine. Infection with Salmonella is characterized by attachment of the bacteria to cells lining the intestinal lumen. Salmonella selectively attach to M cells, which are found in the intestinal mucosa. The bacteria are then internalized by receptor-mediated endocytosis. Salmonella multiplies within a vesicle inside the cell. Occasionally, they manage to pass through the intestinal mucosa at M cells to enter the lymphatic and cardiovascular systems, and from there, they may spread and eventually affect many organs. Salmonella bacteria live in the intestines of people, animals, and birds. Most people are infected with Salmonella by eating foods that have been contaminated by feces. Commonly infected foods include raw meat, poultry, seafood, raw eggs, fruits, and vegetables. Feces may get onto raw meat and poultry during the butchering process. Seafood may be contaminated if harvested from contaminated water. While an egg's shell may seem to be a perfect barrier to contamination, some infected chickens produce eggs that contain Salmonella before the shell is even formed. Some fresh produce, particularly imported varieties, may be hydrated in the field or washed during processing with water contaminated with Salmonella. Contamination can also occur in the kitchen when juices from raw meat and poultry come into contact with uncooked foods, such as salads. Salmonella is never considered part of the normal intestinal flora. It is always pathogenic and can cause 4 disease states in humans. First one is typhoid fever. The most virulent serotype of Salmonella S. typhi causes the bacterial disease typhoid fever. Unlike the Salmonella that cause salmonellosis, this pathogen is not found in animals. It spread only in the feces of other humans. Before the days of proper sewage disposal, water treatment, and food sanitation, typhoid was an extremely common disease. Typhoid fever is still a frequent cause of deaths in parts of the world with poor sanitation. Globally, an estimated 21 million cases occur annually, causing tens of thousands of deaths. The patient with typhoid fever suffers from a high fever of about 40 degrees centigrade and continual headache. Diarrhea appears only during the 2nd or 3rd week, and the fever then tends to decline. In severe cases, which can be fatal, ulceration and perforation of the intestinal wall can occur. Before antibiotic therapy was available, a mortality rate of 20% was common. With the treatments available today, it is less than 1%. Second one is carrier state. The strains of Salmonella responsible for causing typhoid and paratyphoid fevers are maintained by human colonization. Chronic colonization for more than 1 year after symptomatic disease develops in 1 to 5% of patients, with the gallbladder being the reservoir in most patients. Third one is septicemia. All Salmonella species can cause bacteremia, although infections with Salmonella typhi and Salmonella paratyphi more commonly lead to a bacteremic phase. The risk of Salmonella bacteremia is higher in pediatric and geriatric patients and in immunocompromised patients. And the last one is gastroenteritis. Gastroenteritis is the most common form of salmonellosis in the United States. Symptoms generally appear 6 to 48 hours after the consumption of contaminated food or water, with the initial presentation consisting of nausea, vomiting, and non-bloody diarrhea. Fever, abdominal cramps, myalgias, and headaches are also common. Symptoms can persist for 2 to 7 days before spontaneous resolution. Prevention from Salmonella. Prevention depends on good sanitation practices to deter contamination and on proper refrigeration to prevent increases in bacterial numbers. The microbes are generally destroyed by normal cooking. Chicken, for example, should be cooked to an internal temperature of 74 degrees centigrade and ground beef to 71 degrees centigrade. Diagnosis of Salmonella. Diagnosis usually depends on isolating the pathogen from the patient's stool. Serotyping is used in outbreaks to identify strains. State public health labs get a DNA fingerprint of isolates. These fingerprints can be used to track an outbreak to its source. Do not forget to like the video and subscribe to our YouTube channel.